we'll be discussing the ethical framework for clinical trial selection along with the practical processes and decisions. So I'd like to welcome clinical ethicist Preisz and paediatric neurologist Helen to the webinar. And following their presentations, it's an absolute pleasure to welcome Kris Elvidge from the Childhood Dementia Initiative, who will be joining the panel to participate in the discussion and answer selected questions. So please submit your questions throughout the webinar via the Q&A tab in the Zoom controls. And if you see a question submitted that you would also like to have answered, you can upvote the question by clicking the thumbs up beside that particular question. So without further ado, let's begin with Anne, our clinical ethicist at the network. Thank you so much, Anne.
So hi everybody, and thanks so much Paula and the Kids Research Institute for inviting me to reflect with you on some of the ethical considerations of clinical trials in paediatric rare diseases. And today, while I will be highlighting ethical values you'll all be familiar with in your everyday work, like avoiding harm and maximising benefit, respect for autonomy and justice, I also want to incorporate and distill those familiar concepts initially into 3 values: proportionality, or balancing exciting, much needed progress and arriving at a net positive when balancing burdens and benefits; precaution, which is sort of hastening carefully and reflectively into the future of novel technology and research, with children's interests firmly at the centre; and protection, both of vulnerable cohorts of children and their families, and also, really importantly, of clinicians and researchers venturing into these novel areas of research with leaps in knowledge previously unimagined in human history. Finally, I want to circle back to add another value, and that's prescience, and thinking about future-proofing these processes and protocols for research in paediatric rare disease. I'd also like to acknowledge that this is Reconciliation Week today, and I'm on the land of the Darug people, and wherever you are working or residing today, I would like to acknowledge the profound and long-standing culture, connections and custodianship to the land of Aboriginal people, and also honour their elders past, present and emerging. I express hope for solidarity and unity for the future health of all children, those with rare diseases and especially First Nations children. So in terms of some general reflections, I want to just ask what makes research ethical in general. I'd like to also start with a cautionary bioethical tale in thinking about the Willowbrook studies briefly, move to ethical values for paediatric context in clinical trials, and ask whether should children be considered ethically different, and particularly those with rare diseases, and then move towards fair trial selection and support for families and patients and clinicians, and Helen will be taking over from there. So what makes clinical research ethical in general? It seeks to promote social and or scientific value and scientific validity or plausibility. It has a level of equipoise where a hypothesis is proposed and the outcome is not certain. Fair subject selection, which means fair, open distribution and recruitment procedures of trial places, a favourable risk-benefit ratio, and embedded processes for independent review with built-in design, monitoring and appeal processes, as well as an informed consent or assent process in the case of children. We also have to think about respect for persons and potential and enrolled subjects. And this is especially relevant for children unable to make their wishes known, whether through their age, their development, or an intellectual disability. There are over 8,000 rare diseases affecting between 3.5 to 5.9% of the population, with 70% of those commencing in childhood. So approximately 6,000 affecting children. And they include, but are not limited to, such conditions as spinal muscular atrophy, rare paediatric cancers, Duchenne's muscular dystrophy, childhood dementias, and many other genetic related diseases and metabolic diseases. So these conditions are simultaneously rare, and yet they're everywhere. 1 in 12 babies is born with a rare disease, and 30% will die before 5 years. Many rare diseases, despite recent advances in genetics and genomics, have no treatment. Early, rapid and accurate diagnosis and options for therapy are crucial and can really improve outcomes for these children. So are children ethically different? Children are unique and they're vulnerable. They're not just little adults, and clinical trials have historically been unavailable, even more so for rare diseases. So in the 60s, the term therapeutic orphans was coined, And rare disease, in a sense, makes them actually doubly orphans. And we have often relied upon extrapolation of adult data, and we continue to do that. The principle of respect for persons, which in adults could be called respect for autonomy, is really important, as many of these children are voiceless, and promoting their voice is not always easy. So they require a suitable proxy and should not be disadvantaged by enrolment and the risk that must be low if there's no prospect of direct benefit to them. But progress for rare disease is crucial, and to treat those diseases or conditions at the molecular level could also profoundly benefit more common conditions. The 1989 Convention on the Rights of the Child really recognises that the right of the child to the enjoyment of the highest attainable standard of health, including the right to have research evidence for treatments commonly used in children, and children with rare diseases must be included in this account. It's a reasonable requirement for the development pipeline of new interventions to include children in trials when their use in children is anticipated. Early phase trials are more ethically acceptable in children with severe or life-threatening conditions when there is no proven treatment or when standard therapies have failed. And these trials require appropriate preclinical safety and efficacy data available from animal studies, modelling or other predictive studies. Yet there continue to be deficiencies for paediatric cohorts, including inadequate funding for trials, and the choice of progression of trials is still being driven by financial and political factors. I want to briefly look back to a precautionary tale about the Willowbrook studies, which occurred at a New York institution for intellectually disabled children between 1956 and 1971, and they were run by a well-known researcher called Saul Krugman. And he felt strongly that it's his duty and our right to perform an experiment on man whenever it can save his life, cure him, or gain him some personal benefit. And these at the time were really groundbreaking studies, and he was a really respected ID specialist. And these led to the discovery of Hep A and B and a gamma globulin vaccine. And children already at Willowbrook were injected with hepatitis antibodies, an experimental group, and others were not, so they were the control group. New students were injected with antibodies and observed to see who developed hepatitis. And this was done with consent of the parents. And the study was sanctioned by the Epidemiological Mental Hygiene Review Board, so that's sort of HATRAC the day. So well children were exposed to harm with the justification to parents that they would get hepatitis in care anyway, but without full disclosure of the study protocol, and insufficient segregation meant that hepatitis spread rapidly through the institution as there was no knowledge of the carrier. And this led to a lot of questions, including about normal or an intellectually disabled child when no benefit is necessarily available for that child. These experiments, which were really on vulnerable institutionalized children, brought up the moral concern that children who are institutionalized should not be used for human experimentation, and that institutions have a duty first and foremost to the patient's well-being and to improve the institution and not to turn the institution's failings to experimental advantage. The public outrage from this study actually led to publishing the Belmont Report, which is now crucial in the underpinnings for all our understandings of research and the differences particularly between research and therapy. And it's a comprehensive guideline of basic ethical principles that guide modern clinical trials. And I raised this case and study not as a reminder that trials may have inadvertent harms to subjects, we all know that, but really to reflect on the fundamental shift in the socio-cultural view of what was considered ethical at that time, and to propose that we seek what might be called epistemological humility, or being humble as we seek new knowledge, whether it be through trials, academic research, or other ways of knowing. And, you know, the fellow who actually was the doctor at the time who, at the end of these studies, said, I'm not wanting to crucify Krugman. Hepatitis B is an international disease that there's now a vaccine for, but let's never, ever do that again. So how should we future-proof clinical research? We should never assume our morals are superior to those of people of the past, because we will indubitably be judged in our turn by our descendants, who may think it abhorrent, for instance, that we allowed children to have mobile phones or Instagram accounts, All these elements are crystallised into socio-cultural societal values, and in a pluralistic society, we really should seek to be open, fair, treat like children with like, and perhaps rebalance ethical weight when there are structural inequities that implicitly underserve certain groups, such as, for instance, Aboriginal peoples. I want to now turn to the values I've distilled. Over time, we've really evolved to a view of protecting children from research to one of protecting children through research. And I guess in terms of proportionality, I want to highlight that progress is important, but also judiciously including children in research and maximising the benefits and minimising the burdens to them, weighing both their physical and psychosocial well-being of the child and the family, and maintaining some equipoise in the goals of research versus therapy. In terms of precaution, we have to be careful about dual agency awareness, where there's transparency regarding the potential for harm and understanding substantial risk in rare disease trials, thinking about how important co-design is and fair access, and considering how we ameliorate reinforcement of structural inequities. As far as protection, I've already mentioned respect for persons as patients and subjects, and promoting access of patients and families to valid information about their condition, to understand that dual agency is a factor, and to differentiate realistically between research and its goals as compared to that of therapy, and also collegiality, and to offer support for clinicians making challenging resource decisions with their known patient cohorts, and having a mechanism to eliminate non-financial conflicts of interest. So just diving deeper into protection, I've already mentioned that children are unique, and I think as far as rare diseases, commonly there are few available options, and they've often exhausted standard therapies, and medicines are generally off-label or unlicensed anyway. There's also the fact that these trials and the factors around these cases are often emotive. Rare diseases are often tragic and severely life-limiting, and they affect multiple family members. So some child designs may demote the chances for siblings to access trials when viral vectors are involved, for instance. And there are also few valid studies generally for many reasons, including wishing to protect children from harm of unknown side effects. But the other factor is industry funding, and cost recouping for paediatric research is low. So costs are recouped only with proof of safety and efficacy in early phase trials, and compassionate or expanded access schemes, which would create implicit pathways with many ethical challenges, especially in rare diseases where children are afflicted, parents are desperate, and clinicians' professional values lead to heartfelt hopes for therapeutic options. Yet the level of evidence is often low due to lack of available or valid studies, and there are unclear trial endpoints when time is still of the essence. So the question here is there greater ethical obligation to intervene early with potentially life-changing interventions for children? And I would suggest that there's certainly weight in that. We're also, as far as protection, there is the duty to care for patients as subjects, and that's primary also for clinicians as researchers, where this line has become increasingly blurry between the clinic and research. And this is underpinned by the increased challenge of a well-known and cared for group of patients and a profound acknowledgement of the tragedy of their situation. There's also consciousness and clarity required regarding dual agency, and by that I mean distinguishing the aims and purposes of known treatments versus the non-therapeutic aims of trials that may not ultimately benefit that child themselves, but may benefit the cohort more widely or the health community. Allowing for and mitigating against the potential for therapeutic misconception, or what you might call misestimation, where the hoped-for benefits of a trial unconsciously demote risk and underpin ethical values. So awareness of that is inadvertently favouring perhaps a likeable family or a more behaviourally compliant patient for trial recruitment, for instance, and mitigating that can be a really important element of future-proofing. So I've used the term solidarity here to think about how we might support stakeholders, and I don't mean by that just patients, because I think they're primary, but also the families that support the children and the clinicians themselves. So all stakeholders in this collaborative partnership moving forward. I've used the term here to think about that because we need to come up with mechanisms and ongoing mechanisms to support colleagues, whether it's through external review committees, external support, collegial support, or communities of practice for decision making, but also support when things might go wrong, when there's a potential harm from a trial or a child even dies. We need moral spaces for support and sharing clinician concerns and cares, and also that for families, such as social workers, psychological support, and a community approach. We need to partner with families in co-design and co-production, and also advocacy groups acknowledging that in rare diseases, their expertise is really important to ongoing understanding of the natural history and perhaps also endpoint outcomes. So from inception, this can be a really important element of developing future-proofing, and we need to share that expertise together. This painting is called The Judgment of Solomon, and here Dice brings to the viewer to the painting to elicit the profound distress inherent to the scene where a mother is giving up her child rather than allowing Solomon to adjudicate on whose child the mother is. Sometimes determinations like who is eligible for a so-called scarce resource, but which is intrinsically a choice about many equally deserving children in often tragic situations, can seem an impossible one, or often what we call in ethics a world of competing sorrows. So how should clinicians, researchers best determine which children should be eligible for rare diseases in clinical trials when these are known cohorts of long-term patients who they care about deeply? And I think this painting elicits that difficulty of those choices. I want to just quickly move to talking about justice, the substantive justice, which is made up of procedural fairness and distributive fairness. And I put this under the umbrella of protection, as it's particularly relevant in terms of a scarce resource, like a clinical trial place, but also maintaining openness and transparency about how we approach clinical trials for rare diseases. Promoting respect for children as persons is as regards to choice and not excluding any child unfairly based on assumption or unexplored bias that can sometimes be structural in terms of the system, and acknowledging those challenges as far as Aboriginal or children or other groups that may have issues in even accessing a hospital or an institution to have follow-up monitoring, for instance, or even knowledge of potential trials, and the ability to advocate for a child, as we know, should not be solely based on medical literacy or socioeconomic advantage. So thinking about ways to mitigate that is a really important part of justice. We also have to understand that the potential benefits may be to the health community, not to the participant of the research, and that there is a potential that they may experience harm, but it must be minimal for the case of the child, and that others may benefit from this trial or other trials, and we must seek to answer a significant scientific or public health question about the health and welfare of children. And further, there has to be transparency of progressive of these trials, the information, and that other studies may rise and may sequentially improve outcomes. Finally, international justice for families is also really an important element of justice. Interactional justice is a really important element for families, and that means clear communication, being able to access the information, accessing their care providers during trials, before trials, and after trials. As terms of precaution, I just want to raise that some of the early phase trials are clearly clinically transformative, and we have to still think about who is the cost bearer. There is a risk of overestimation of potential benefit by affected families and an underestimation of substantial risk. In 2020, $20 billion flowed into the biotech companies, and high development costs and the need to incentivise innovation are common justifications for high costs. But how public funding played an important role in each product's invention. Zolgensma cost $2.1 million US and is now PBC funded or publicly funded, and Luxturna for ocular gene therapy is approximately $475,000 US. And this is not to downplay how clinically transformative they are for children, and also that those costs can quickly be annotated given the costs of ongoing disease burden and treatment in the affected. But it remains ethically relevant to retain transparency and cost oversight as more and more gene therapies emerge and the biotech sector grows rapidly, which with many new startups offering great promise and hope, but also with risks if they fail. Industry development companies primarily answer to their shareholders, and cell and gene therapy stocks rose about 25% in 2020. So novel gene therapy should be fairly priced in order to promote equitable access for patients and sustainable reimbursement for payers. The other thing I just want to mention is the importance of data and privacy. And this could be a whole seminar in itself, but it's about data collection and reporting, not just for safety, but also efficacy. And this is more important with gene therapies and newer classes of therapeutics intended for patient populations that may be quite small and also may be especially vulnerable. And there has to be precaution regarding privacy and children's genomic data, which can be linked and potentially affect future options for a child who has not themselves agreed to be part of a study and may not understand how their information will be stored or used in the future. So this is the proceedings of a workshop in 2020 that looked at clinical trial design and considering the emerging ethical and clinical questions of that. And here parents were often used as surrogate decision makers for their child, and they have often multiple and conflicting values. And this was one of the things that came out as a serious concern for parents particularly, and that trials require the child and a parent to stay at the clinical site for approximately 30 days. There's multiple concerns about that, and I know Helen's going to touch on that, so I won't spend too much time here. I just wanted to highlight that there are a number of ethical issues that were distilled, and that they basically mean that patients and families should be part of research development process, and that clear, iterative and open communication can really build trust, and sufficient time and summaries of information can really help to make decisions that can benefit participation, and that families can suffer really very high indirect financial and non-financial costs, including disruption to the family unit to participate in trials. So we have to find ways to support them, and this study on this workshop showed that families really do find value in having a support system available comprised of research nurses, physicians and contact with other families and peers who have gone through similar situations in research trials. I want to just consider a few questions here, and these came out of the trial, and I won't spend much time on them, but they're to do with things like, are there alternatives to placebo in a clinical trial? Are there alternatives to exclusion from a trial based on a previous exposure to, say, viral vectors or other parameters? How do we equitably consider siblings? And Helen's going to look at this. Is it possible to know in advance that how therapeutic a gene therapy will be? How can we transparently and consistently educate the patient and the family about expectations? And how long will it take the field to get to these particular rare diseases? All questions which I think we may not have had time to answer today, but are really important. So as Mandela said, history will judge us by how we treat our children, and especially those with rare diseases. So future-proofing our protocols and protecting children now and in the future is in everybody's interests. Protecting families and clinicians as investigators is inherent to that aim, but we must also ensure that changes in societal mores don't leave us with the same moral regret that the Willowbrook studies did at that time. Only then can we learn from the past to inform a better future for the children we care for. So I've included in this prescience, which really means future-proofing and developing an ethically defensible operating protocol for trial inclusion, and considering children now and maintaining an open future for them in the future. So I'm now going to say thank you, and I'm going to hand over to Helen to take us into the next phase of our discussion about these ethical considerations in trial selection and design.
So that was great, Anne. Thank you so much. And I'm actually going to talk in a little bit more detail about one specific aspect of ethical problems around clinical trial access, and that is equitable access to competitive clinical trials and the issues that we have around participant selection. So to just give a bit of background to this, I work in the neuromuscular clinic, and so my experience of this is around the DMD gene therapy trials. So just to focus on this quickly, and then I'll broaden it. Duchenne muscular dystrophy, or DMD, is an X-linked neuromuscular disorder which causes muscle weakness, disability and premature death. Gene therapy via the AAV vector is seen as a promising and potentially curative option for a really devastating disease. But gene therapy is still in the trial phase and has a significant risk profile. There are 3 potential gene therapy trials in the pipeline for DMD at SCHN, but there are very limited trial spots. And a patient's question at a recent parent forum really sums up the question that I'm trying to ask today, which is, if you have 8 eligible boys and 3 spots, how do you decide who gets the treatment? So what do you do when there are more eligible participants than available trial spots? So I'm going to quickly just talk about the ethical principles and values that underpin fair participant selection, and then the options that we have for participant selection, talk a little bit about what others have done, and then I will talk about our proposed allocation procedure, which is very much a work in progress. And then I'll speak a little bit about affected siblings. So the relevant ethical values and principles around fair subject selection: firstly, justice. Justice is fairness. So we want to ensure equity of opportunity, and also that the benefits and burdens of any research trials are shared amongst the community. Beneficence is maximising benefits and minimising harms. Respect for persons, we want to ensure autonomy and protection of our vulnerable patients, and also ensure that our participants in trials are representative of the whole population to give our studies social and scientific value. And then finally, and very importantly, transparency. So we want to ensure that eligible participants are aware of available trials and the method of allocation. So what makes clinical research ethical? Anne's already talked about this a lot, and this is a paper from Emmanuel in 2000, but it's still pretty relevant. I like this slide because it's all fits on one slide. 7 requirements for what makes research ethical, and I'm going to focus in on this one, which is fair subject selection. So selection of subjects is really, we need to make sure that stigmatized and vulnerable individuals are not targeted for risky research, which Anne talked about in the Willowbrook studies. But also the flip side of that is that the rich and socially powerful are not favored for potentially beneficial research. So when we think about considerations for fair subject selection, we need to have specific inclusion and exclusion criteria which are related to the scientific goals of the study. We need to avoid recruiting participants based just on convenience, and I mean by that researcher convenience or vulnerability. Groups and subjects should not be excluded from access without good scientific reasons, and involving diverse patient populations ensures generalizability of research findings. And as I said, justice demands that the benefits and burdens are equally shared by the population. And this paper from Douglas McKay makes a good point in that persons do not have a right to be enrolled in a trial, but persons are moral equals and as such should be granted equal opportunity to access trials. So all prospective subjects who meet the scientifically defined eligibility criteria and for whom participation is consistent with their medical interests should have formal equal opportunity to participate. So with that in mind, if we think about what the options are around subject selection when we have very limited trial spots, we could do it on a first come, first served basis. We could use targeted selection or random allocation. Those are 3 of the options. So if you think about first come, first served, in this trial spots would be offered to eligible patients who approach the team. And this is a very pragmatic option and has been used to date quite successfully, but the evidence does suggest that factors such as socioeconomic status and education level will affect trial engagement. So the risk of this model is that you will get a very skewed population with reduced generalizability of results. So another option is targeted recruitment, and again, there are many proponents of this method of allocating spots. So patients are chosen based on a physician's subjective view of their ability to participate and adhere to the demands of a trial. So it's cherry picking patients really. And there's a potential here to reward patients for earlier trial participation or for good adherence to treatment. But there are risks with this, that of implicit bias, that is unconscious negative evaluation based on irrelevant characteristics such as race, gender, or language barriers, because researchers do share the biases of the general population, and this can lead to ascertainment bias, which reduces the scientific validity. This also risks increasing structural barriers to access, and Anne talked about this a little bit, increasing health inequity, because increased effort is required to recruit culturally and linguistically diverse patients and those who are socially disadvantaged. And this can, if we don't put that effort in, this can lead to reduced generalizability of results. So the third option is that of random allocation. And in this, spots are allocated randomly to eligible patients. And the benefits of this model are that researchers are obliged to overcome structural barriers to access, which improves equal opportunity and generalizability of results. The risks of this is that certainly more effort is needed to assist those with reduced resources access trials, and it also risks wasting highly valued trial spots on someone who is unable to complete the trial, which is a valid concern. So in this space, when we look at it, what have others done? This is a paper from 2021 on equitable subject selection in concurrent COVID-19 trials. And the authors commented that COVID-19 created an unprecedented need for life-saving therapies with multiple clinical trials all happening at once. Fair subject selection then became an urgent moral concern, as there was a moral risk if wealthy, educated, and the powerful got enhanced access to potentially therapeutic trials. So this team developed some recommendations around fair subject selection. Firstly, physicians must accept the role of neutral medical advisor and acknowledge the uncertainty, the risks and benefits. So as Anne said, avoiding therapeutic misestimation. Ensure eligible participants are aware of all ongoing trials. Research groups have a duty to disclose alternative trials. And clinical trials should adopt an allocation principle and communicate to all patients and healthcare workers what that is. The allocation principle should include either a blinded system or a scoring system to avoid implicit bias. And then this paper from the Journal of Cystic Fibrosis from Rebecca Dobra, this is quite an interesting paper because it was a similar situation to where we found ourselves with the DMD gene therapy trials. So the rapidly changing cystic fibrosis landscape, there are many more willing and eligible patients than available slots in these trials. So the aim of these authors was to offer equal opportunity to all patients meeting inclusion or exclusion criteria to minimize conscious and unconscious bias and ensure generalizable results. So they developed a standard operating procedure to ensure equity and allocation. And this is the procedure. It's really quite simple. So the inclusion and exclusion criteria are fulfilled. The list generated for patients in a random order. Patients were then contacted in order to see if they would like to participate and could participate. If they could commit to the timelines, screening visit was planned. If they didn't want to participate or couldn't commit to the timelines, the next patient on the list was contacted, and so the system moved on. The benefits that they found from the standing operating procedure were firstly that it was transparent, so everyone was aware what was going on. It minimized selection bias, improved equity of opportunity, and it also importantly mitigated against potential challenges and complaints. And they also made the point that it opened recruitment to patients who otherwise would not have been considered by the team. So this slide shows some researcher and patient feedback quotes on this standing operating procedure. From the researchers, the first time it was used, the entire group selected to screen had not participated in a commercial trial previously, and this included a teenager with autistic spectrum disorder, who the authors admitted they would not have approached to enter the trial, but he proved to be an excellent trial candidate. And this from a parent: no one picked our son to have CF and no one picked him for the trial. Both were acts of random selection, and he's doing really well. And importantly, I think from a patient who missed out on selection, to not be selected is upsetting. However, if you're not selected, you can take comfort that somebody else is getting a chance and you can understand why they got that chance. So these authors then went on to actually survey the CF community about what they thought about their standard operating procedure and how it had worked. So they did an online survey of patients, parents, and physicians. And interestingly, 75% of them agreed that all eligible participants should have an equal opportunity in recruitment. There were some conflicting responses, with a preference for prioritizing patients with a good adherence record, which is the targeted recruitment. And interestingly, 80% said that they would move to a different location if that was necessary due to a national allocation of spots. But what they all thought was that transparency was essential, whatever method of allocation was decided. So if we come back to our group, this is what we have come up with as a draft, a proposed operating procedure for gene therapy within our group. The aims of this are to ensure equity of opportunity, minimize selection bias, ensure benefits and burdens of research are shared, overcome the structural barriers and reduce health inequity if possible, and promote generalizability of results. We also want to ensure transparency. We have not yet got family or advocacy group inclusion and input into how we do this, and that's something that we want to do. And we haven't incorporated a model around clinician support, which is something else we would like to do. But this is where we are up to. So the clinical trials will be discussed with the patient cohort at clinic appointments and all eligible patients included in a general pool. We then decided we would select patients in a random order, so go for the random model. Selected patients would then be reviewed by a panel of clinicians, nurses, and allied health. And if all eligibility criteria are met and there are no really compelling reasons why we would not progress, the patient would progress to the next stage. The clinician then meets with the family to discuss the trial risks, the level of the commitment required, and also the other side of that is what assistance the family may need to enable them to participate. If the family and patient remain interested and they proceed to formal consent and screening, including antibody testing. And if the patient and family decline, we move on to the next randomly selected patient. And the whole process is a movable feast, which should be under review and evaluation. So that's where we've got to. And as I said, we would really welcome feedback from the audience about what they think about this. So finally, I just want to say a brief word about siblings. What we're talking about here are genetic disorders. So there's certainly the possibility of affected siblings, and we certainly have affected siblings in our clinics. And if one child is randomly selected or selected in another way for a trial, and the sibling is not, there are significant risks with that. And the risks include psychological distress for the family, survivor guilt in the treated sibling, and also viral shed by the treated sibling infecting the untreated sibling and rendering them potentially ineligible for gene therapy in the future without draconian isolation requirements separating both siblings. So it's a difficult space. One option is expanded access or compassionate access programs could be considered for siblings, but they come with their own risks. And I think reading through the literature on this, what's coming forward is that a view that pharmaceutical companies need to get involved with this and that they should develop transparent procedures for managing expanded access requests. They don't necessarily have to agree with the expanded access requests, but it should be clear what their procedure is around this. And I think that is a question that maybe needs to be asked of the pharmaceutical companies that are approaching the hospitals. So that's me for now. So I might just hand back to Anne, and she's going to just sum up where we're up to, and then we'll take questions.
So I guess just summing up, I think in thinking through this, and thanks Helen for that great presentation, we all have an ethical obligation working in this area to promote safe, effective and innovative research with children and families, especially for those with rare diseases and few options. There should be a prospect of direct benefit to the child's health and an important scientific public health question pertinent to their health and welfare, and the possible risks and benefits have to be weighed in that context. We need to protect and ensure fairness for vulnerable children, and more so, again, for those with rare diseases. But we need to future-proof robust, fair procedures and trial design as societal mores and ethical standards may change. And I'm just referring back to the Willowbrook studies and the shift in societal understandings of what consent means for families, how we manage perhaps coercion or sense of coercion. And we need to have transparency, which should underpin all interactions with multiple stakeholders in clinical design and fair trial inclusion and process. We also need to think about how we feasibly provide support for patients, families, and also clinicians who are investigators, because as this gap between therapy and research closes, this is going to be more and more important both before, during and post the trial process. So thank you, everyone.
Thank you both Anne and Helen for those really wonderful presentations and for really setting the scene and framing our conversation today. And I noticed we have already had a question submitted, but before we begin the discussion, I'd like to welcome Kris Elvidge to the panel, who'll be providing us with a patient and family perspective. So I'll just hand over to Kris so that she can introduce herself to the webinar. Thanks, Kris.
Yeah, so I am the head of research at the Childhood Dementia Initiative. I have a scientific background, but I left the lab about 15 years ago to start working with patient organisations, firstly in muscular dystrophy, both in the UK and in Australia. And then I worked at the San Filippo Children's Foundation for about 5 years, and the past 2 years with the Childhood Dementia Initiative, which is advocating for the more than 70 different types genetic diseases that cause dementia in childhood. And with all of these genetic diseases under our umbrella, all the issues that have been brought up today are certainly things that we are seeing in our community. So it's such great presentations today, bringing to the forefront the issues that we see quite often, especially missing out on places in trials and not really knowing why. The issues with AAV antibody testing and then isolating or not isolating or testing positive and then missing out, and also the issue with siblings, and if one sibling gets treated and the other doesn't. So I'll hand back to Paula now.
Thanks, Kris. Excellent. Okie dokie. So thanks, Kris. And I'd like to now welcome Dr. Laura Fawcett, who is a respiratory consultant at Sydney Children's Hospital, Randwick, and Dr. Michelle Lorenzos, who is a paediatric neurologist from the Children's Hospital at Westmead. And they will kindly facilitate the Q&A session today. Thanks.
Thanks, Paula. We can see a couple of questions coming in, but we thought we would let Kris introduce the first question, because we know she's got a list to ask our panelists.
Yes, I've got lots of questions. I want to learn a lot from today for our community as well. So what I'm interested in knowing is what support is currently available for families going through clinical trial selection and possibly being successful, or if they miss out, what support is available.
I might put that question back to Michelle, because she's our actual clinical trial leader. And so she's the one who's on the forefront of what's happening with that, because we're not really at the stage yet of starting these trials. So I might put that back to Michelle.
And I might also add, I'm curious to know if there are gaps and if that's something that the patient organisation could be working on or advocating for.
So thanks, Helen, for bringing that question back to me. So I'm one of the neurologists who's also working in the space of some of these clinical trials, as well as other non-gene therapy related trials in neuromuscular medicine, and clinical trials lead across our hospital. So I think that's an excellent question, and one that we talk about a lot. So I think the first thing is that we are trying to reflect on the experiences of families that have gone through the clinical trial process by listening, asking for feedback and evaluating their experiences. And we're doing quite a bit of work in understanding their journey. And then moving forward, first of all, we work very hard around the communication space and the patient-clinician communication space from the moment anything about a clinical trial is mentioned, making sure that things are as transparent and clear as possible, and that there's frequent opportunity for questions and second opinions, and we facilitate those. If a patient chooses to be taking part in a trial and then they go forth, then our model of clinical trial facilitation, if you like, usually involves a clinical trial coordinator and research nurse who's assigned to a particular patient for that trial, as well as regular contact with the investigators. And we really work hard to make sure those relationships are supportive for the family as well. And then our clinical nurse specialists remain involved, and they are often important in bridging the care that goes before the clinical trial and afterward, and making sure that because they have that prior knowledge of the family. So for instance, some of the things that we've done recently in our clinical trial space is move weekly infusions for a child that started school to be in the afternoon to make sure that that suits his routine better and is easier for the family. I think probably one of the gaps that I recognise is actual dedicated psychological support, and these are very stressful situations for families, whether they're selected or not selected, and we certainly have access to social workers within the hospital, but I think formal recognition of that and support around that is something that we see as a potential space to improve.
Thanks, Michelle. I think we were going to let you ask the next question, Michelle.
That's right, I'll ask this question to somebody else. I might, first of all, actually ask Kris to comment on this, and then perhaps some of the other panellists. But we have a question from the audience about the understanding of risk and the fact that often we have quite desperate families in situations, and we're asking them to partake in difficult decision-making for them and the clinician. And whose decision is it to decide what is an acceptable risk for this child? The child, the family, or the clinician?
Yeah, that's a tricky one. And we know that families' appetite for risk is much higher than anybody else when they're faced with a fatal degenerative condition. So very, very tricky. I think I'll have to hand over to the ethicist.
And maybe I'll have a bit of a go at that. So I suppose one of the things we consider in terms of ethical consideration for trial access is something called the prospect of direct benefit. And so that would sort of take into account what a reasonable parent acting in the best interest of the child would allow regarding his or her child to participate in a trial after weighing the probability of possible benefit and possible risk. And we know that if that direct benefit is not likely for the child, then you need to think about minimal risk, and that's minimal risk to the child. So there is weight to having prospect of direct benefit to a child, particularly in these early phase trials, and that's, I think, a really important element, that weighing of the risk and benefits. But that can't be done solely by the parent alone, because as Kris has so keenly pointed out, that substantial risk will maybe be underestimated. So that is part of how a good trial design is set up as far as the standard operating procedure for recruitment. But also one of the things we haven't talked a lot about is how we embed in terms of procedural justice, a fair process in terms of an appeals process, so that accountability and revisability for an appeals process if a child doesn't get included in the trial, and sort of having some sort of process around that embedded in the inception of the design and of the standard operating procedure.
I think if the family has very clear and they understand the risks and the benefits, and they decide that it is in the benefit of their child and it outweighs the risk, then I think you have to trust those parents.
Yeah, I think sometimes it can get quite complicated. If we think well-publicised cases, not really in the clinical trial space, but in terms of experimental therapy. If we think about little boy Charlie Gard, who was a very well-publicised case from the UK, who had a mitochondrial disorder, and all of the clinicians had decided that going forward with an experimental treatment would cause him really a degree of harm that was intolerable, whereas his parents disagreed. And that went all the way through the courts, and the parents eventually did get to take their child away and proceed with treatment. But it was an incredibly difficult journey for everyone involved, and I think that kind of marks the extreme end of it, when parents and clinicians are really, really butting up against each other over what constitutes significant harm to a child. And in most cases that would not be the situation, and I think in most cases clinicians and parents can reach some kind of common ground about what is in the best interests of the child.
Thank you all. There's another couple of questions around the ethics of having a selection process itself. So there's a comment from Marino who reflects that if there is any child who meets the inclusion-exclusion criteria, that they should be given an opportunity to participate, and having any selection process is unethical. But then also a question around the size of the cohort that might be eligible being a potential impact. So the comments and thoughts on those.
I mean, you know, I would, you know, on one level agree with Marino in that, you know, yes, if you have a trial that could be of therapeutic benefit, then it would be, in a perfect world, we would want everyone who was eligible and wanted to participate to access that trial. But we have to also bear in mind that this is still a trial. This is not a treatment. And that's a different situation from we have a treatment that is available to everybody. So I think those are two things to be, you know, although it's a supposedly therapeutic trial, it's still a trial with significant risks. So that is something to bear in mind. And what was the second part of the question? Sorry.
So the size of the potential cohort might affect the selection process.
Look, the cohorts, I think it varies depending on which trial we're talking about. And, you know, we can't really talk in specific details about each cohort that we have going through at the minute, but there will be, in some of the trials, there definitely will be more eligible participants than spots available. And so that is the point of, you know, it's not a situation that anybody wants to be in, but it's trying to make the best of what we can do with the situation that we have. You know, in a perfect world, we would also have more trial sites and we'd be able to expand this throughout the country and throughout the world, but that's not the situation that we're in.
Yeah, I would just echo that. And just, sorry, go on, Kris.
We'll just say in parallel, we would want to keep working to get more trial sites, more trials, more opportunities for those that may have missed out.
Yeah.
Yeah, and also potentially expanded access when that's available. But we've got to remember that these are early phase trials. And so they're not phase 4 trials, they're early phase, they're still novel. And we have to remember that, and that that balance of, you know, risk and benefit, which is, you know, equivocal, and it has to be determined. It has to be done under some sort of reasonable and structured format that we can actually gather knowledge reasonably. Otherwise, we're making assumptions that actually aren't verifiable.
I think just to add to that, that if you think about some of these trials being early phase, then there are many early phase trials that are stopped after that early phase because they are deemed to not be efficacious or safe. And so whilst we do want to make sure we give access and opportunity, there are sometimes good scientific reasons why we are starting with small populations of patients for scientific rigour and safety. There is another question asking about the inclusion of siblings, mentioning that if you acknowledge that compassionate access might be an alternative, that is there a risk there because it doesn't have the same scientific oversight as a clinical trial, and then how do you balance that?
I mean, I'm happy to talk to that just briefly, but I'm happy for you to jump in as well, Anna. I mean, I think there are risks with compassionate access programs anyway, and because, you know, one of the risks is that if you don't, as you've said, if you don't get oversight of the trial or results, and from that, then there's a risk that people will go down the compassionate access route and get treatments, and then we don't get results, and we don't actually know what's happening in that space, and then that reduces the number of people who will actually be eligible to go into trials if they go down the compassionate access route. But I do think within the sibling space, I do think that there is room for a compassionate access program, because I think that the risks to the family of having discordant siblings is really significant. And, you know, Michelle and I have been in a situation like that with some siblings, and that was a very difficult situation to manage.
Yeah.
I think Kris has some experience of that as well.
I think one of the responsibilities of having children in a trial is not to do further harm to them. That is one of the things I think in terms of ethics, it's about the best interest of the child, but the interests of the child rest within their family in terms of a relational autonomy. So we have to consider this a little bit more complexity when there are siblings involved in a protocol and they may not get access.
Yeah, I know of a family where one child in a family was treated and the other wasn't, and they had to isolate for months after the treatment was given, separating the family while the AAV virus was being shed so as not to expose the other child. And that was really difficult for the family, not only for the parents not seeing their other child, but the siblings were extremely close as well, both having the same disease and really supporting each other normally, and they had to be separated for months, which was really tricky.
All right, we've got time for one more question, a quick one.
Okay, ooh, pressure's on to choose it. Well, the next one that's got the highest votes is from Kirsten, who comments that often with rare diseases, there are more more common rare diseases that will have clinical trials available to them with highly publicized results. But for children who have the ultra rare condition where there's nothing available, how do we balance the hope of what might come whilst being realistic for those individual families?
I'll have a quick go at this. I think this is of course, Kirsten, you've gone straight to the heart of the matter. I think one of the really important things is that, approaching, progressing and innovating about rare disease can actually help, increase the pipeline for innovation for perhaps even rarer diseases. But, that has to be, it can help with common conditions, but it also can fundamentally build on knowledge. And I think we have to also acknowledge the tragedy of some of these incredibly rare conditions for families, and that unfortunately they have been so underserved in terms of research, as children have generally. But if we start to continue to think about, working with children and protecting them through research rather than excluding them from research, I think there is hope for the future that there will be more options for children with very, very rare diseases.
Thank you, Anne. And I think we could probably have another hour discussing this topic, and it's really fantastic to see the questions, and unfortunately we have run out of time. However, we will be providing answers to the questions we didn't have time to discuss today via the website in the coming days after the webinar, so please keep an eye on that, and we'll certainly endeavour to respond to those questions. I would really like to thank everyone for joining us today. A reminder that the video of the webinar will be available via our website and the Sydney Children's Hospitals Network YouTube channel. There will also be a short survey at the end, so if you have a couple of moments, we'd really appreciate it if you could fill that out. And most importantly, I really wanted to thank Anne, Helen and Kris for a fantastic webinar today, and to our Q&A facilitators, Laura and Michelle, and most importantly to you, the audience, for your active participation. I really want to thank the staff at Sydney Children's Hospitals Network who've supported today's presentations. A lot of work goes on behind the scenes, and I want to certainly extend our thanks to them. Importantly, please stay tuned for our next webinar, which will be happening in July. And for July's webinar, we're going to be exploring the patient journey of gene therapy for inherited retinal disease. And we look forward to having you join us for then.